To today. Oh, here we go. Okay. Welcome to today's live stream. Thank you everyone for joining us. I'm Nilofer Farjo, the current secretary of the ISHRS, and joining me today is Professor Maksim Plikus from California. Welcome, Max.
Thank you, Nilofer, for having me here.
Great to see you. So Professor Plikus is a hair biology lecturer, and he's going to be talking to us at the ISHRS 28th Annual World Congress, which is going to be virtual this year in October. So I'm going to just kind of very briefly with Max touch on a few areas of particular interest that he's worked on in the past. And then we're going to briefly discuss the topic that he's going to present at our meeting, which is discussing groups of hair follicles regenerating as a spreading wave. So just before we start the interview, if I can give you a little bit of a brief background. Dr. Plikus has worked with two well-known researchers who've been at our meetings in the past. First with Cheng-Ming Chuong, during his PhD degree in pathology at the University of Southern California. And here he studied, amongst other things, the mechanisms of hair growth control. Later, he joined George Cotsarelis, who we all know very well, at the University of Pennsylvania. And here he did some work on wound regeneration. So now Dr. Plikus is back in California at the University of California, Irvine, and he heads a research lab that investigates key questions in hair follicle biology, including mechanisms in stem cell control and hair length regulation. So at the end of our short interview today, we may have time for a few questions. So if you do have questions, on the right-hand side, you can see that there is a comment section. So if you click on that, you can add your questions as we go along. So let's begin with the first thing that I wanted to talk about. So I want to just kind of get a little bit of background information from you, Max. First of all, about some of the work you've done in the past, because really I don't want to give away everything you're going to talk about at the conference. So in the past, you've worked on different aspects of skin and hair regeneration, and it seems to me that these two things are very intimately entwined in their behavior. So do you want to just maybe give an example of some of the work that you've done looking at these two systems and how they interact?
Yes, sure, Nilofer. I think like you very rightfully said, I was lucky to work with two really great skin and hair biologists, Cheng-Ming Chuong and George Cotsarelis. So I think a lot of my thinking has been heavily influenced by them. And one thing I learned to appreciate is just how intricate and beautiful hair follicle is as the biological model. It's an amazing model for studying things that I care about, so stem cells and regeneration. And another really interesting thing about hair is they do not exist in isolation. They are certainly very intimately interconnected to one another through a variety of mechanisms, and they communicate not only with one another using molecular language, they also communicate with the surrounding tissue. So specifically, they communicate with skin adipocytes, they communicate with skin fibroblasts. New research is emerging showing that they receive a lot of communication signals from skin nerves. So you have to start really thinking about all the hairs on the skin as one interconnected and codependent network. So when upon disease, one thing goes wrong, surely other things go wrong as well. So and that's what sort of heavily influences my research, is those external cues that hairs receive and send, and how things work across the entire skin, not just within one hair follicle.
So you've mentioned adipocyte and adipocyte signaling, which has kind of sparked interest in the hair field in terms of what we do when we transplant. And people are using adipose-derived stem cells as an adjunct to doing hair transplantation, or even as a standalone treatment. But I think a lot of us are very maybe confused is too complicated a word for it, about how this works. And I think as well, which type of stem cells we're talking about,
Yeah.
and from what site. So, we're talking about dermal white adipose tissue. However, people are not necessarily getting their cells from the correct location. Do you want to just maybe briefly comment on
Yeah,
that?
this is a really great point. I mean, as all of us know, this buzzword, mesenchymal stem cells, or adipose-derived stem cells, is very popular. And these are fibroblast-like cells that seem to have stem cell-like properties, and they are relatively easy to isolate, for example, upon routine liposuction procedure. So it becomes very tempting, because these are patient-specific mesenchymal stem cells, to graft them into other sites where you can hypothesize they might have beneficial effects on things such as hair. But I think you correctly pointed out that we do not really fully understand what are we grafting when we're talking about adipose-derived stem cells. It's most certainly very complex and not purified population of cells. It contains a lot of fragments of blood vessels. There are true adipocyte progenitors. There are also other mesenchymal cells. There are vascular cells, endothelial cells. So all of this stuff goes in when we graft it. And the other really interesting question that truly remains unanswered: do grafted cells feel happy when they are taken out of inguinal fat or subcutaneous fat in the abdomen and placed into the scalp? Because as far as we understand, all cells in our body have what we call molecular code, meaning there are certain types of molecules that tell cell where do they belong. And if you graft them, they know, in the way they know, that they're in the wrong address. And so they feel like a foreigner in a foreign country without able to speak foreign language, not so comfortable. So there are outstanding questions. Can these cells survive? Can they really positively contribute to the biology of the new tissue? So can they positively contribute to hair biology? We don't know those answers.
Right. Yeah. So do you think that's really very interesting? And do you think that stemness and that would apply to what we do in hair transplantation? So moving occipital hairs to the frontal scalp. So, for instance, we do see that over time, those hair follicles that have been moved do change. So we often get patients 20 years later who come back and they say, oh, well, their hair follicles that were very robust and their hair transplant that looked very thick at the time that it was done has now started to thin and doesn't look the same anymore. So do you think there is that happening as well with these hair follicles?
And I think, what you're saying is a really great insight. I think the dogma in hair transplantation was that occipital hair remain testosterone insensitive, even if they are grafted into otherwise testosterone sensitive recipient area. And I think from my reading of hair transplantation literature, for the most part, that remains true, that the epithelial and dermal papilla cells in the grafted follicles retain the original biology. But as you said, and as I said, hairs are also acquiring local signals from the surrounding cells. And perhaps in the beginning, after transplantation, most of the signals are acquired from the adipose cells that were carried over with the graft. But as years and decades pass, the host tissue becomes more integrated with these hairs. And it's very possible that the host adipose tissue in the frontal skull is also testosterone sensitive and secretes what we can define as negative factors. So over time, you might start seeing those additional kind of inhibitory effects from outside of the hair in the host tissue now affecting the grafted hair. So this is quite possible.
Right. Well, that's really a very fascinating point, because it is something that in our discussion groups that we've discussed and amongst ourselves, are other people seeing it? And everybody does see it. So that's really actually a fascinating insight for us. Anyway, we can talk forever on adipocytes and all that, but I just wanted now to maybe just focus a little bit on the actual talk that you're going to give at the conference, just to give people a flavor of what you're going to talk about. And you've been working with a mouse model and the connection with the human hair growth cycle and it regenerating in a wave-like pattern. So do you want to just briefly give a little bit of an insight into what you've been working on, and then maybe, how that might relate to humans, because humans aren't mice. However, our genetics are pretty much identical. If I'm correct in saying that.
Yeah. As many in the field, in basic biology, we work with mice, and I would say mouse is a good but far from perfect approximation of a model to a human hair. So there's a lot of human hair biology aspects that are not replicated in mouse model, and that's the limitation. But at the same time, there are many fundamental aspects of hair biology that we seem to share with mice. And one thing that we studied in mice, this is going back to my PhD days, and we continue to do work in this area, is that hairs do not grow individually. They grow as groups. And actively growing hairs could actually send signals to the non-growing neighbors. And those signals, we understand part of their molecular makeup, actually can trigger the non-growing hairs to grow. Now, those hairs start to grow, they start to send the same signals to their neighbors. Their neighbors get triggered to grow. And what you get in the end is a chain reaction. So when you look at shaved mice or other types of rodents, you would actually see that hair propagates as a moving wave. It's like a really physically moving wave. And if you start taking pictures of animals and you assemble them later, you stitch them together, it becomes very clear that there's this beautiful propagating wave of hair growth. And what this really tells us that in the population, at the population level, hairs have support mechanism when actively growing hairs stimulate their neighbors to grow. Now, if we can somehow tap into that, and if it actually exists in humans, at least to partial extent, then we can actually have a mechanism for hair-to-hair growth stimulation. That existence does not require exogenous molecules to be added. So the big question was always, is it there?
Yeah, and do you think the opposite might occur as well, that if a hair follicle is switched off, that signals to the growing hair to then start switching off?
I would definitely not exclude that possibility. I would definitely not exclude the possibility that hairs that just stop growing, they could produce inhibitory molecules for hair growth that could affect their neighbors. So this population level wave-like hair growth behavior, it's very obvious in rodents. It's very obvious, for example, in rabbits. But in humans, the question always was, is this even relevant? So as far as we understand, and everybody could agree, is that we don't see patterns and waves of growth on a scalp of a typical human. But from human embryology and neonatal biology, we actually know that at newborn stage, many babies that are on month 3 to 4 months old, they actually display really curious phenomena. I think medically it's defined as transient neonatal alopecia. So it's kind of a synchronous fallout of the hair from the occipital area to a point where babies all of a sudden develop transient bald spot that goes away as new hairs grow. I think most people incorrectly assume this is because baby always lays down on the pillow, and usually actually babies should not have a pillow, just laying down in the crib, rubbing the head. The true cause of that is actually synchronized exit from onogen, synchronized exit from hair cycle, and as a consequence, synchronous fallout of the hair. So we do have rudimental wave behavior, at least in newborn stage. So if that could be somehow reawakened, then we might have a completely novel approach to hair growth stimulation.
Right. And do you think the embryological difference between the hair follicles at the back of the scalp and the top of the scalp also are relevant as well?
Yes, I do think that obviously hair follicles in different anatomic sites are quite different. And we know that, in fact, from mice. So if you actually don't only concentrate on studying back mouse skin, but you actually start looking elsewhere, as we did recently. So we looked at the ventral mouse skin, the facial skin, ear skin. We actually realized that hair biology in those sites is quite distinct. And for instance, we published that ventral hairs intrinsically cycle at a much faster pace, but they can communicate with the back mouse hair. But because they cycle faster, what you start seeing is this body-wide waves of hair growth propagating from the abdomen to the back in mice. So essentially, ventral hair sort of works as kind of a pacemaker of hair growth for the rest of the body. So what we understand is, although distinct, hairs in adjacent skin regions can talk to each other because they share the core molecular language. So they do understand each other's molecules. So dominant hairs in the ventrum can actually stimulate hairs in the dorsum. Similar could be happening in human skin at different sites.
Right. Okay. Okay. So I'm just looking to see if we have any questions for you from the group. So there's one question: is there one key signal that you believe is more important than Wnt or BMP?
Yeah. I mean, great question. I'd love to have, one for all solution, but everything that we understand about the hair to date is that it's so much more complicated that we assumed that there's certainly not one signal. And I think sort of the dominant concept in biology right now, as we now have those unbiased approaches to study cells and tissues, is that there is not a single molecule. Everything works as a network, what we call gene regulatory network, where thousands of genes are intimately intertwined, delicate jigsaw puzzle. Just killing off one is not enough to kill the entire system. Just adding one molecule is not enough to completely reboot the entire system. So it tends to be more complex like that. But that's the challenge ahead for us biologists.
And then, of course, there's the whole topic of epigenetics, environmental factors, and it goes on and on and on and on and on. And when I got into hair, as most of us as hair surgeons, we're from a doctor's point of view, we didn't know any hair biology. Certainly I didn't know anything about hair biology, and I thought, oh, this is a very straightforward, simple procedure we're doing, and eventually someone will come up with a cure for baldness, and then end of story. But it's turned out to be a very, very fascinating journey for me, learning about some hair biology. But believe me, I really only understand a very fraction of what you guys are doing in the lab. You're the real intellects, and we're the ones who are just, people say we're just moving hair about. We're not, but in very, very simple terms, that's kind of what.
I could say the same. We only study mouse hair, being completely ignorant of the mouse to human differences. So But I think there's a positive side to that daunting complexity, is that there's more than one solution. There's clearly more than one switch that you can manipulate, and perhaps manipulating more than one switch at the same time can have really synergistic effects that we currently do not anticipate. So I think some of the major discoveries in hair biology, honestly, are still to come.
That's great, and that's good to know. So I would like to thank you on behalf of the ISHRS and the Virtual Congress Committee for taking the time out to be with us today. And I want to remind the audience that you can watch the live streams done by other guest speakers. They're already available, and this one will be available shortly. And we will have some more live streams in the run-up to the Congress. And if you want to use the meeting hashtag ISHRS 2020 to share these events on your social media, that would be great to promote the conference. And most importantly, please don't forget to register. Early bird deadline is coming up, September the 30th. And for that, please go to 28annual.org. And I really, really enjoyed talking to you today. We've hosted hair research meetings, but we've actually not spoken to each other in person. And I'm glad we had this chat today. And looking at the comments, I've got comments from people all over the world who've been watching today. And I hope those people will have some really good questions for you at the Congress. So I'd like to thank you for that. And
Nilofer, yeah, thank you very much for inviting me. It's really great pleasure. I look forward to the meeting. It'll be a great opportunity for me personally to learn more about human hair biology from all of the attendees. So really quite excited. Thank you so much.
Great. Thank you, everyone, and goodbye. See you soon.
Thank you, everybody.